So with that, we'll get started. I want to start with, let's see. Yeah, I mean, of course, there's a standard disclaimer. Now that I work for a corporation, again, all the material in this presentation is my personal opinion, does not reflect the views of either Pfizer or any other organization that I'm affiliated with. Right. I want to start our session on a slightly philosophical note, right? And this is something that we always believe is kind of, three guiding principles that we need to have when we think about running clinical trials, right? And the three guiding principles, and you might have gone over some of these yesterday as well. I apologize if it's a repetition, but we start with something which is ethics, right? And ethics has to be the bedrock of all clinical research, right? And when we talk about ethics, we talk about three things. We talk about respect, we talk about beneficence, and we talk about justice, right? Um, and if you think about it, what is respect? Respect is, ensuring that every clinical trial participant deserves our respect, right? So you're exposing them to maybe new therapies, new schedules, new indications, new ways of administering the drug. You're collecting data from them, and you want to make sure that the data that you collect is done in a manner which is absolutely respectful, right? We've heard about things in the past where either prison inmates were subjected to experimentation without them knowing, Black Americans were subjected to experimentation without them knowing, and so on, right? So respect is absolutely critical, right? Beneficence is essentially, as a clinician, your main objective is to ensure that the patients are always benefited, whether it is through the new drug or it is through the existing drug. So as a clinician, every clinical trial has a possibility to basically say, I don't think this drug is working well at all. I don't think it is providing benefit to the patients. And so I want this patient to get off the clinical trial, right? So that should always be something that you have available. So beneficence is absolutely essential, right? And the third is justice. Make sure that everything that happens in the most fair manner that's possible, right? So these three principles should be our guiding principles towards the ethics part of why we do and how we do clinical research, correct? The second one is transparency, okay? And all of you who have been in the field know about things like informed consent, about patient information packages, all of those kinds of things. Be absolutely factual about what you're planning, how you plan to conduct the study, and how you report. Yeah. And reporting is probably one of the most important aspects. Even if the clinical trial is negative, even if it does not achieve its primary objective, it is essential that we go ahead and say and report it actually for what it is. Yeah. And so I'll give you an example of where, we might be well-meaning, but we might end up doing the wrong thing. Yeah. So there was a study that was conducted in India, by government and nonprofits around, around vaccination schedules, right? And what happened was they were trying to see if there was a different schedule, easier schedule that was as effective as the existing, let's say, more complicated schedule for vaccinations. Yeah. And what ended up happening was that they wanted to enroll, let's say, 300 patients in the study. Okay. They tried doing it for 5 years, and they could not enroll more than 60 patients into the study. Okay. Imagine where 300 is, imagine where 60 is. Okay. And so what they did is, at the end of the study, at 60 patients, they actually found no evidence that the new schedule was better than the old schedule. Okay. But that's not what they reported. What they reported was that the new schedule is equivalent to the old schedule. It's not inferior to the old schedule. Okay. Now, think about it. It's a slight change in exactly how you're reporting it, right? On one hand, you were unable to show your primary objective, which is that the new schedule is better. But then what you actually reported was the exact opposite, which is to say, that the new schedule is not inferior to the old one. And Dr. Shrikant is going to talk to you, over the next 2 days about superiority and non-inferiority and so on. But the whole idea here is that the interpretation for these two things becomes completely different. On one hand, you're saying you are having an experiment to show that the new one is better, but because you are unable to show that, you can't report that the new one is not worse than the old one, right? Because they're two separate things. And so, it is absolutely critical that you're factual about your planning, about your conduct, as well as about your reporting. Yeah. We want to ensure that every step is taken after due process. You've thought this through, and anybody who reads this tomorrow, they have to have confidence. That you have to have confidence. And somebody had once told me that they use a mother test, right? Whenever they're running clinical trials, which is the first one is, are you running the clinical trial in a way that you would be comfortable with enrolling your mother into the study? Yeah. Are you providing information about the clinical trial in a way that you would expect your mother to understand what is happening in the study, right? So always try and use, they were saying the mother test, which is if you can convince yourself that your mother understands everything that you're talking about the trial, that you feel comfortable about the conduct of the study, that, your mother can, you would enroll your mother in the study, then you're fine. Yeah. Then the clinical trial is generally okay. Yeah. And the third one is the third kind of bedrock, the third principle is scientific unbiasedness. Yeah. And this is absolutely critical. When you start a study, you have to start a study with knowing perfectly well that you don't know the answer to the question that you're trying to solve through the clinical trial, right? It cannot be that you run a clinical trial to show something that you already know. Yeah. The clinical trial has to say, I don't really know this, and so therefore I want to be able to answer this particular question. So that is how you design a study. Yeah. Unbiasedness. And that allows you to interpret correctly when the results come out. Because otherwise, if you already think that one is better than the other before you start a clinical trial, then ethically it's not okay for you to even run the clinical trial, because one is actually better than the other, and you have to do the right thing by the patients. Yeah. So the ability to understand the results, accept the results of the study, hinges on your unbiasedness. Yeah. So unbiasedness gets into a lot of things. We have guys have talked, I think, about randomization yesterday. That's one way to ensure that it's unbiased, and so on, right? So how can you have both internal validity and external validity? How can you generalize, and how at the same time can you have internal validity that shows that the design that you have selected, the study that you've done, is actually valid? Yeah. So these are the three basic principles, and it's tremendously important for us to remember this, because every aspect of the clinical trial that we do from a statistical standpoint ends up affecting one of these three or more than one of these three principles, right? So if you take hypothesis testing, which you will learn about later, it's about scientific unbiasedness and transparency. If you talk about confidence intervals, it's about scientific unbiasedness. Yeah. If you talk about a sample size, it impacts unbiasedness and ethics and so on. Yeah. So a lot of the clinical research, the statistical principles and methodology that you'll be using hit more than one of these principles. But the one that we're going to talk about today, design of experiments, it actually impacts all of them. Yeah, it impacts ethics, it impacts your unbiasedness, and it impacts your transparency. So therefore, it is critically important that the way you design your study, and what we're going to talk about today, is critically important to ensuring that the clinical research that you do is valid, and the clinical research that you do is something that passes the mother test. Yeah. So those are a couple of slides that I wanted to start off, which talks a little bit about why is designing a study so critically important, right? So I just want to maybe move on to the kinds of designs that we use, but before that, a quick check, to stop and see if there are any questions, any comments about what we have spoken so far.
Do any of the participants have any questions?
Is the pace okay? I mean, is it too fast, too slow? Are you guys able to hear me okay?
Yes, sir. It has been clear so
Okay, all right. Okay, good. So let's move on to, so we're going to talk a little bit about phase one designs and phase two designs in this session. Phase three designs are so complicated and so large that I think Dr. Shrikant and Dr. Jaiseelan are going to talk about that over the course of time, right? Over the course of the next day and a half. But let's start with phase one trials, and we'll start with phase one trials in the non-oncology setting, and then we'll move to the phase one trials in the oncology setting. The slightly different, the slight differences between the two, right? So in the non-oncology setting, the objective is to mainly find if the drug is tolerated and does not pose an immediate safety risk. Yeah. And you also use these to sometimes figure out a range of doses that may be feasible that you can take into a phase two setting and then use a phase two setting to actually figure out the right dose. Yeah. So in the non-oncology setting, the phase one trials are typically done in healthy volunteers. And when I say non-oncology, I mean like, most of the non-oncology, non-rare diseases kind of thing, which is life-threatening, right? Where the medicines, the drugs that are used are so toxic that you don't want to expose healthy volunteers to those kinds of drugs, right? So that's what I mean when I say non-oncology settings, right? These designs are typically very, very straightforward. So they either have something we call a single ascending dose, which is that each patient gets only one dose, yeah. But then the next, so you expose one group of patients to one dose, make sure it's tolerated, then you expose the next group of patients to the next higher dose, and so on. So each patient or each subject, in this case, each healthy volunteer, gets exposed to only one dose. Yeah. Or you can have multiple ascending doses. Yeah. So which means that you have one cohort, they start with one dose and you see it's well tolerated. Then you have those same people move on to the next higher dose to see if that is also well tolerated, right? So you can either have a what they call a single ascending dose, or they can have a multiple ascending dose, right? Of course, if this is a first in human study, then you have special care, right? I mean, it's typically done within a phase one unit. You're asked to be in the unit for a period of 2 days, 3 days, a week at a time, sometimes, and everybody's looking to make sure that once they give you the new medicine, that everything is okay and you're doing fine, you're able to tolerate that. Yeah. So these are typically the kinds of designs that you have in the non-oncology setting. Right? So fairly straightforward. And the whole objective is to ensure that the drug is reasonably well tolerated and that the drug is, and that you start to have some basic idea of what the efficacy of the drug is likely to be. What are the good dose levels where the drug starts to show an effect? And most of the ways that you start to do that is through pharmacokinetic, pharmacodynamic parameters. Okay. So what are PKPD studies? I think there is a separate, again, session on PKPD later on. But PKPD studies are where, you give people a particular dose and then you keep drawing blood at specified time points. And then you start to, plot and see how does the drug behave in the body, right? So what is the absorption? What is the distribution? What is the metabolism for the drug? When is the maximum time that the drug, what is the time when the drug reaches maximum concentration? What is the maximum concentration? All of those kinds of things, right? So you start to plot all of this and you use that to model what could be the best dose for the drug. Yeah. So that's typically what you tend to do in a phase one setting for, the non-serious kinds of therapeutic areas. Okay. Now, when it comes to oncology or other types of therapeutic areas like oncology, which are life-threatening, these trials are typically done in patients. Okay. And the main reason is because many of these drugs are so toxic, right? I mean, think about most of the oncology drugs, right? I mean, so far, now you get slightly different kinds of drugs, but mostly so far, they are what we call cytotoxic, which is they go and kill the cancer cells. And it's very hard for them to differentiate between cancer cells and regular cells in the body, right? Because they have many of the regular cells in the body have similar parameters, like the cancer cells, like for example, the hair cells, yeah, or the cells in your gut, yeah. And that's the reason you see that when cancer patients go through chemotherapy, the hair falls off, right? Because it's hard to differentiate between that and a cancer cell. They typically have a lot of nausea, vomiting, all of those kinds of GI toxicity, because again, it's hard to differentiate between that. So these drugs go and attack these other cells in the body as well. And so therefore, you don't want to expose healthy volunteers to these kinds of medicines. Yeah. And so they typically are done in patients. Uh, in the majority of the cases, the phase 1 trials are not just to assess safety and tolerability. That's a big part. But it is also to get to the right dose, because it's very important in these kinds of studies that you get to the right dose as quickly as possible. And because you're doing it in patients, it gives you an opportunity to get to the right dose. Yeah. And so because so far oncology drugs have been majority cytotoxic, the goal is to find a maximum amount of drug that the body can tolerate without it being too toxic. Yeah. And when we say too toxic, it doesn't mean like, nausea, diarrhea, those kinds of toxicity, because that's kind of what they say, accepted toxicity, right? Too toxic here typically means that you have a grade 3 liver toxicity. Your liver starts to, go bad, or, you're hospitalized, or, you have really, really bad, grade 2, grade 3, like very severe events. And those are the kinds of events that are deemed too toxic for some of these, very, let's say, life-threatening diseases, right? And so the idea in a phase 1 study in oncology is typically to make sure that you're able to give the patient as much drug as they can tolerate. Yeah, good. So we're going to start with, basically, this is a definition slide, right? And we will get into the details of how we come about some of these things. But this is basically a definition slide just to get us all on the same page. So as we said, the objective of a phase 1 oncology trial is to find what we call the MTD, which is the maximally tolerable dose among a set of J doses. Okay. And what we do is we expose patients in a sequential manner. And generally, the way we expose them used to be something that we called, of a Fibonacci sequence. Okay. So basically you start with 1, but then you go 1, then you go 2, then you go 3, you go 5. So many of you that know what a Fibonacci sequence is, it's the addition of the previous 2 numbers. Yeah. So that is how it used to be. Okay. These days, there are slightly, different ways. You have something called a modified Fibonacci and other kinds of things. So what you do is that down below that you see these set of doses. By the way, can you see my pointer, my arrow? Yeah, okay, good. Okay, so you see these set of doses. These are not Fibonacci sequences. Okay, they basically are, you're going to double until a certain point, and then after that, you're going to go, for a set of doses. So from 1, you more than double to a 2.5, you double to a 5, you double to a 10, and then you're going to go 15, 20, 25, 30, and so on. Yeah. So this is a different kind of a sequence, not really a Fibonacci sequence, but you try and set up this number of doses. Okay. And then your idea is you want to try and give patients the dose that is maximally tolerated. Okay. You try and figure that out. You collect the data, which is called a DLT, okay, or a dose limiting toxicity. Okay. What that is it is an adverse event, okay, like a grade 3 liver function failure or something. Yeah. It's an adverse event that limits the dose that you can give. Okay. So that's why it's called a dose limiting toxicity. Yeah. And what you collect is a binary variable. Do they have a DLT? Yes or no. There is no gradation in the DLT. It's not like it's a mild DLT, it's a severe DLT. There is no such thing. Okay. DLT is either you have a DLT, yes or a no. Okay. And obviously, you think about your safety data. Your safety data is really much, much more high dimensional, right? You have grades in the toxicity levels, you have severity, you have relationships, you have all of these different things. And in a way, what you do in this study is you reduce all that dimensionality to this just as one parameter. Do they have a DLT? Yes or no? Okay. But unfortunately, in a lot of early stage clinical trials, that's the best that you can do. Okay. And you will see that there's a lot of statistical methods that are used to make this as useful as possible. Okay. So those are just the definitions for now. Yeah. And what you're looking for is what is the probability of a DLT? So what is a DLT rate? Okay. So at a particular dose level, what is the probability that you will find a dose limiting toxicity? Okay. And let's call that, let's say PJ over here. Yeah. Which is probability of having a dose limiting toxicity DJ. And what you want to do is you want to keep this PJ, this probability of having a dose limiting toxicity, to a certain number that you feel comfortable with. Okay. You can't have a DLT of 0 for some of these oncology studies. Is impossible. Yeah. Because if your DLT is 0, means that you are dosing people at a level that's not even going to impact their tumor. So the efficacy is also going to be very, very low. Okay. So you have to accept some level of toxicity. And most of the times, you kind of keep the toxicity level for oncology studies between a 6 and a third. Okay. So 0.16 to 0.33. That's typically a DLT level. Yeah. So you keep your probability of DLT at like either a 25%, 20%, 25%, 30%, and so on. Okay. So whatever that works for you for that particular study. Okay. And then the design is typically that you start with a very low dose, which is a safe dose, okay, which is extrapolated from preclinical data. So you have animal models and other kinds of things. You extrapolate from that, get a dose that is really low, right? And then you keep going up, as you know, through the levels that we spoke about. And we escalate, which is we go up to the next dose only if it is reasonably safe. Yeah. Good. So that's the general principle. Okay. So now we move on to what we call, so there are two kinds of designs that one can use in these phase 1 studies. Okay. You can either have a very rule-based design. Okay. So examples of those are 3+3 type of designs. And we'll speak about that a little bit. Okay. Or what they call simple up and down design. So you go up, if you get a DLT, you come down and so on. Okay. So those are the simple base and they are called algorithmic designs. Okay. Because they follow a particular algorithm. Yeah. The other kind are called model-based designs. Okay. So these are designs where, you know, they're adaptive in nature. And what you do is you actually get information from the previous doses. Okay. And then you model it. So you keep modeling the dose limiting toxicity based on accumulating information. So you can either do it in a CRM kind of a method, okay, continuous reassessment method, okay, which has a Bayesian flavor, or you can have a proper Bayesian design, okay, like a Bayesian design, which is called a Bayesian optimal interval design, okay. Or finally, you can not just look at, you know, safety, but you can also incorporate efficacy into the mix, and you have what are called the TEPI designs, okay, toxicity and efficacy probability interval designs, yeah. And we will quickly go through over the next like 30 minutes. We will go through these designs and see how these designs work. Okay, and we'll come back to the slide later on where it talks about, you know, which designs are better and so on. Okay, so with that, we're going to start with our first sort of traditional design, which is called the 3+3 design. Okay, and the 3+3 design is one of the oldest designs, but continues to be very, 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 you know, popular even now. Okay, and what it does is that the name comes from the fact that you enroll 3 patients at every dose cohort. Okay. So let's say you start with the first dose cohort. You know, remember the first slide that we had where we had our first dose cohort at a level of 1 milligram, right? So you start and put 3 patients at 1 milligram. Okay. And then you evaluate these 3 patients for typically about 1 month. Yeah, 28 days. That's the typical time that you evaluate these patients. Okay. And then you look to see how many of those 3 patients have had a dose limiting toxicity. Okay. If none of them have had it, so at 1 milligram, 0 out of the 3 patients have a dose limiting toxicity, then you can go to the next higher dose level, which, you know, if you remember was 2.5. Okay. And then you add 3 patients on that level. Okay. If, for example, if instead of 0, let's say you have 1 out of those 3 patients who had a dose limiting toxicity at 1 milligram, what you do is you add 3 more patients, which is the reason it's called 3 plus 3. Yeah. You add 3 more patients at the same dose level at 1 milligram again. Okay. And then you look to see what happens to them. Okay. If now out of 6 patients, out of the 3 additional patients that you see, already 1 out of the 3 had a DLT. If out of the new 3 that you've added, if none of the new 3 have a dose limiting toxicity, so your total numbers of dose limiting toxicities are only 1 out of 6. Out of 6 patients, only the first one that you added, I mean, the first out of the 3 that you added, only that person has a DLT. The new 3, none of them have a DLT. So your DLT rate is 1 out of 6. Okay, what you do is you go to the next dose level. So you escalate again. Okay, but if out of the remaining 3, the new 3 also, okay, if 1 or more has a DLT, okay, then you stop. Okay, you stop here, and what you say is that the dose level before this, now there is no dose level before 1, so the experiment has stopped and you can't continue the drug at all. But if you have gone beyond 1 to 2.55, 10, and so on, okay, then the dose level before that, where you have already passed 0 out of 3 or 1 out of 6, that dose level is called the maximum tolerated dose. Okay, so this is what you do if you get 1 out of 3 in the first set of 3. Okay, if in the first set of 3 itself you get more than 1, so either 2 or 3 patients, all of them have DLTs, then again you do the same thing. You either go to the next dose level, lower dose level, or you declare MTD at the lower dose level. Okay, so you escalate if you have either 0 out of 3 or 1 out of 6. Okay, if you have 1 out of 3, you add 3 more. If you have more than 1 out of 6 or more than 1 out of 3, then you stop and you either go down or you declare the lower dose level to be the MTD. Okay, make sense? Yeah. Any questions on this? No? Okay. Yeah. All right. So this, this design, even though it seems very, very simple, has stood the test of time. In fact, I remember, I think it was 2-3 years ago, I was at CMC Vellore and I was having some discussions with Professor Jayasilan, because we were trying to do this kind of a design for a snake bite study. And I think there, instead of a 3 plus 3, it was a 10 plus 10. So we were looking at 10 patients at a particular dose level of the antivenom for a snake bite. Yeah. And we were looking at a 10 plus 10 design, but same exact concept. Okay. So, so it is amazing. So if you look at this particular design example, so we have already done this, right? So let's go through this one more time. For cohort 1, 0 out of 3 at 1 milligram, so no worries. Go to cohort 2, 2 milligram. Okay, 0 out of 3, no problem. Go to cohort 3, 4 milligrams. Okay, so 1 out of 3, right? So what do we do? If we get 1 out of 3, we add 3 more patients, correct? So the fourth cohort is now 6 patients. We still have 1 out of 6 only. So out of the added 3, nobody got a DLT. So you move on to the next cohort, which is you add 8 mg. Yeah, and at 8 mg, you get 1 out of 3. Okay, so you add 3 more patients to 8 mg. It ends up being, you get 1 more DLT in at 8 mg. So you have 2 out of 6. So you go back, you stop, and you declare 4 mg to be your MTD. Okay, so same thing that we talked about before. Okay, and the amazing thing is that even though this is such a simple design, okay, it is used a lot because it has a lot of nice properties. Okay, so for example, it has been shown that if you continue to keep on doing this over a long period of time and over many, many studies, the the operating characteristics, right? So this design typically gives you a dose level with about 30 to 33% toxicity. Okay, so those are some of the nice long-term properties of this design. So therefore, it is a design that is still very popular and used quite often in the industry. Okay. The problem though is that, let us say that these are, this is what you're, this is what's going on. Okay, so let's say that, similar to what we did before, 0, 3, 0, 3, 1, 3, 0, 3, so you escalate 0, 3. Okay, you go up, you get 1 out of 3, 1 out of 3. Okay, and let's say you de-escalate and you get 1 out of 3 over here. Okay, what ends up happening is that you declare this dose level 4, okay, to be your MTD, even though you have had only 1 out of 6. Okay, in this case, you have had 1 out of 6, but you escalated. But in this case, you have 1 out of 6, and you never escalate because the next dose level is already at 2 out of 6, and you declare this to be your MTD. Okay, so it's a fixed design. No matter what happens, you have to follow this design. And by doing this, what ends up happening is that, look at this, okay? The blue line here is your probability if your true DLT rate were 0.5. Okay, this is the blue line. Okay, is what happens if your true DLT rate was 0.5. Okay, and you actually realize that if your true DLT rate is 0.5, okay, 11% of the time, 1 by 6, go all the way up. 11% of the time, we will see either 0 or 1 DLT in 6 patients. So even if your drug is actually very, very toxic, even if the DLT rate is as high as 0.5, the chance that you will see either 0 or 1 out of 6 patients having a DLT is as high as 10%. So 10% of the time, you will end up having escalating for a drug which is too toxic. Okay. On the flip side, if your true DLT rate is actually 0.166, 26% of the time, you will see at least 2 DLT in 6 patients. Okay. So even if your true DLT rate, okay, is as low as 1 in 6, 25% of the time, more than quarter of the time, you will actually end up seeing more than 2 DLTs. Okay, in 6 patients. Yeah. And so you end up stopping thinking the drug is too toxic, but it's actually not too toxic because your DLT rate is only, to true DLT rate is only 1/6, 0.16. Yeah. And so there is a very high probability here, 10% of the time, you will end up finding a dose is acceptable when actually it is not. 25% of the time, you will end up finding a dose is too toxic when actually it is not. Okay. So you end up, 1 out of 4 times, you'll end up with a dose that's not optimal. Okay. Because you're already thinking it's too toxic, but it's actually not too toxic. Yeah. And so those are the reasons why, these, if you're, these days, you don't see this being used as often as in the past because you've got much better designs that, reduce these probabilities and give you a much better estimate of the true DLT. Okay. Sounds good? Good. So I'm going to talk a little bit now about, other sort of, model-based designs. So one of them is, as we said, is the Bayesian optimal interval design. Okay. And these lambda E and lambda D that you see here, these are the probabilities for escalation and de-escalation. Okay. And so what you do is it's like a 3+3 design. Okay. But what it does is instead of having a fixed algorithm of 0 out of 3, 1 out of 3, 1 out of 6, and so on, okay, the observed toxicity is actually compared to a table with dosing decisions like this. Okay. And so therefore, what you do is that this table keeps getting updated as you start to get more and more information. Okay. And so therefore, this allows you a lot more flexibility. The 3+3 did not offer you any flexibility at all. This is like a 3+3, except that you compare the number of DLTs not to a fixed 0 out of 3 or a 1 out of 6, but in fact to the number of DLTs that are allowed for a table that gets updated based on some Bayesian models. Okay. And that's what we call the Bayesian optimal interval design. Okay. And then we have a design called escalation with overdose control. Okay. And we'll see a little bit more about this later on, where what we say is that, again, we update the model with two parameters. One is that, based on your safety, you can, you fit a model for your dose curve. But also you have another constraint, which is that the true DLT can never be more than 0.5. Okay, that's called overdose control. So you add a constraint to the model to ensure that you never overdose a patient, okay, so that it doesn't become too toxic. Yeah, okay. And then the CRMs and MTPs are all similar kinds of designs. What we should do is we should let us go into one of these designs in a little bit of detail. Okay. And so what we're going to use is we're going to use something called a Bayesian logistic regression model design, BLRMs they're called. Okay. And the underlying model here is a logistic regression model for your dose toxicity. Okay. We're going to use Bayesian criteria. How many people there are familiar with the Bayesian criteria? Okay, so I don't see too many hands up, right? Okay, so let us then talk a little bit about what this Bayesian inference is. Okay. So let's say that, okay, let me ask the question, okay, which is that when you toss a coin, okay, what is the probability that the coin, that you're going to see a heads at the top of the coin? I'm waiting for somebody to answer from the class.
The consensus is 50%.
Okay, so 50%, right? So the first time you toss a coin, you expect that it's going to be a heads with 50%. Yeah, probability 50%. Okay, good. Now let us say that you toss a coin 10 times, okay, and you get 6 heads and 4 tails. Okay, on the 11th toss, what do you think is the probability of getting a heads on top?
50%.
40?
60.
60%. Okay, all right. So I'm not talking about the proportion of heads you've gotten so far. Okay, it's a new toss. Okay, so I'm going to toss a coin new. Yeah. What is the probability that the next toss, the 11th toss, is going to be a heads? And you think that's going to be 60%? How many say it's going to be 60%? Just put your hands up. I can see the class on my camera. So how many people in the class seem to think it's 60%? Okay, I see a couple of hands up. All right. And how many of you seem to think it should be 50%? It's still 50% for heads. Okay. So a lot of people putting their hands up for 50%. Yeah. How many think it's neither 60% nor 50%? Something else. Okay. One half hand up, I see. Okay. All right. Okay. So that's the classic, question about the two philosophies that we have in inference, okay, called frequentist inference and Bayesian inference. Okay. What frequentist inference says is that the coin has an inherent, has an inherent probability, okay, of being of heads or tails. Okay. And if I were to keep tossing the coin, if I believe that the coin is unbiased, okay, then I keep tossing the coin up, okay, 100 times, 1,000 times or something, then at some point, I'm going to get to a place where it will be roughly 50%. Okay, so if I toss it infinite number of times, okay, at the end of infinity, when I check how many heads, how many tails, it will be 50%. Okay, that's the frequentist paradigm. Okay, and so what you do is that when you do your own clinical trial, okay, you basically say that I am going to take a coin and I'm going to, toss it, let's say, 25 times or 50 times, okay? And if I get 26 heads and 24 tails, okay, then my probability is 26 out of 50, okay, which is roughly, let's say, 0.52, okay? And then I'm going to do a hypothesis test to see if 0.52 is different from 0.5, okay? And if my p-value is very small, then I will say that I conclude that the coin is not 0.5, okay, it's not unbiased, so the true rate is not 0.5. Or if the p-value is very large, then I'll conclude that I have now concluded that the coin is unbiased and the true rate of heads is 0.5. Okay, this is the frequentist method of thinking about stuff, of inference. Okay, so a lot of the analysis that we do, okay, is all frequentist. So whenever you go in and say what is the probability of a response rate, okay, and is the response rate for treatment A bigger than response rate for treatment B, all the hypothesis testing, everything that you do is all frequentist. Okay. On the other hand, what Bayesian philosophy says is that I have an underlying belief. Okay. And let's say my underlying belief is that the coin is unbiased with a rate of 0.5. Okay. Now I'm going to toss the coin 10 times. Okay. And if I get 6 heads and 4 tails, okay, then I'm going to update my underlying belief based on the information I've seen. Okay. So if my underlying belief is very strong, that is what we call an informative prior. Okay. My underlying belief is very, very strong. The coin is unbiased. Then even if I see 6 and 4, the data is not going to dissuade me from that belief. Okay. All right. On the other hand, if my underlying belief is very weak, okay, which is actually I don't no idea what the thing of the coin is. It could be anything from 0.1 to 0.9. Then when I see 6 and 4, then I will update my belief and I will say that actually the true proportion of heads, the true probability of heads is closer to 0.6 and not really 0.5. Okay, so the amount of let's say credibility or strength of evidence that the data has depends on also on how much data I collect and how strong my prior belief is. Okay, so even if, let's say if my prior belief was very, very strong that it's unbiased, it's 0.5, if I toss the coin 1,000 times, okay, and I get 600 heads and 400 tails, yeah, now you might start to think, you know what, maybe the coin is not biased, right? Because I'm expecting 500, 500. If I get 510, 490, it's still okay. Even if I get 550, 450, it might still be okay. But 600, 400, that's too much evidence from the data, right? Contrary to my belief. Or 700, 300, that's really, really contrary to my belief, correct? So I update my belief based on what I see from the data. Okay, get it? Now, the funny thing is that medicine by nature is Bayesian. Okay, when you practice medicine, you do it in a very, very Bayesian manner, right? So let us say that at the beginning of, the flu season, okay, somebody comes to you as a clinician and says, I got a cold and sniffles and some fever, you're going to think it's a flu, right? And you give them flu medicine and nothing happens to them, yeah, and they don't get better, and then you do some testing and find out it's something else, it's typhoid or malaria or something, okay? And then the next patient also comes, and the next patient also comes, and by the time the fourth patient comes, and you've treated them for flu, but they don't get better, and you test them and they start to show malaria symptoms, okay? The fifth patient, you're already updating your belief, right? That even though it is flu season, looks like I've already seen 4 malaria patients, so I want to make sure that I test for malaria right in the beginning or something like that, correct? So what happens is you start updating your belief very, very quickly when you start to see emerging data, even though you have lots and lots of data that shows that during flu season, you're going to get 1,000 flu patients. Yeah. But the minute you see a few malaria patients, you start to update your belief, correct? So the practice of medicine, in my opinion, is by nature Bayesian, but a lot of the work that we do in a clinical trial setting is frequentist. Okay. So here, what we have started to do is in phase one, and Bayesian philosophy is a very old philosophy. Okay. It's been around for ages. Yeah. 1700s was when Reverend Bayes, who actually was a practitioner, also was a statistician and a mathematician. He came up with the Bayes theorem in 1700s. Yeah. So we've been using it since then. But these days, because it's a little complicated to do Bayes theorem and Bayesian analysis without strong computing. Now, because our computers have gotten so much stronger, we are able to use Bayesian methodology a lot more. Okay. So what we do is that we use Bayesian kind of philosophy in doing this dose limiting toxicity and figuring out the right model. Okay. So we start with what we call a prior, which is either uninformative or weakly informative, right? We spoke about that. What is informative prior? I know the coin is unbiased, it is 0.5 versus a weakly or a non-informative prior that says, I don't know, it could be anything between 0.1 and 0.9, correct? And the priors are typically based on historical data, okay? And then, to make sure that we really take care of all the data and all our previous thinking, we sometimes make the priors a little bit more complicated. So we have what we call mixture priors, okay? And then we use these two criteria, as we said. One was overdose control, right? We spoke about EVOC, overdose control, escalation with overdose control, which is less than 25. And an example of overdose control is the probability is less than 25% that the true DLT exceeds 0.33. So we always make sure about that. Okay. So we have an overdose control in place. Okay. And then the main thing we want to do is figure out a dose that maximizes the probability that the true DLT is in that interval of 0.16 to 0.33. Okay. Makes sense? So what we start to do is we fit a model. Okay. So this is my logistic regression model. Okay. For those of you who have not done logistic regression, forget about it. It's a model. Okay. So actually, we'll just skip the slide. Okay. And this is my prior distribution. As you can see, there is not a lot of information. Okay. I just feel that the median is around 0.1. So around 0.1 is about 50% of the data. Okay. The rest of the data is around here. So the probability that it is less than 0.1 is very high. The probability is greater than 0.1 is low. Okay. Yeah. So this is a median about this is a prior that I have about my probability. Okay. As we thought about before, we had a probability of 0.5 for the coin being unbiased, right? So that was point probability, which means all the mass, all my probability was exactly at 0.5. So at 0.5, it was 1 and 0 everywhere else. Okay. That's called a point mass probability. This is called a continuous probability. Okay. So the chance it's 0.1 is highest. Okay. Less than that, higher. Lower than that, higher than 0.1, low probability. Okay. All right. So now when I start with this as my prior probability, okay, and it's called a weakly informative prior. Okay. Now I see my data. And what do I see in my data is out of 6 patients that I have treated, okay, one of them has a dose limiting toxicity. Okay. So now if one of them is a dose limiting toxicity, what is my probability of DLT? Is 1 by 6, correct? Which is 0.166666, whatever, right? So approximate it to 0.17. Okay, so you see 0.17, right? What you see here in the parentheses are what are called Bayesian credible intervals. Okay, it's like a confidence interval. So think about it like a confidence interval. Okay, so you see now that even though this was my original thinking, this dotted line, as soon as I see 1 out of 6, my new thinking is that the DLT curve is like this. Okay. It has the highest probability at 0.17, but it's got probabilities on the other 2 sides as well. Makes sense? So what I've done is I've started with a prior probability curve, and then I've updated that based on my data, which is 1 out of 6 patients. Clear, everybody? Okay. Right. Yeah. Sorry, is there a question? No? Okay, all right. Okay, yeah. Yeah, Dr. Tabu, yes.
Why is the uninformative prior that particular shape as opposed to a flat probability of that is doesn't say that you have any information?
Right.
Isn't it suggesting that low doses are bad if you have
Correct.
a particular?
Correct. So the main reason for that, Dr. Shrikant, is, I mean, one, what we try to do is, we try to use a conjugate prior, right? So we use a beta for a binomial so that it becomes easier for us to calculate data, right? So that's a beta with 0.1. And, the main reason again is that, we try to spread the variability out a lot so that, it's fairly non-informative. So even with 1 out of 6, you start to see the shape that you would expect to see. Yeah. Good. All right. Perfect. Right. So now that I see this, okay, now this is my, what I see as a new curve, right? That I've seen. This is my new, what we call the posterior density. Okay. So prior is my prior density. After I see the data 1 out of 6, I get to see this posterior density. And this posterior density, you see 3 regions. The gray region is anything which is less than 0.16. Okay, remember what our DLT region was supposed to be between 0.16 and 0.3. That is what we are interested, right? That is where we want our toxicity to really be. So anything less than 0.16, we think we are underdosing. Okay, so that's gray. So that we are underdosing there. Anything in green is our targeted toxicity. Okay, so this is probably where we want to be in the green area. Okay, and anything which is red is greater than 0.3, so that is overdosing. Okay, so you see now what the probabilities are. The probability for, at this particular dose level with 1 out of 6, the probability for overdosing is this area in red, which is 17%. So we're okay. Yeah. The probability for target toxicity is 35%, which is good. Nice big chunk. Okay. But the probability for underdosing is 48.3. It's very, very high, right? So with this particular dose level and seeing 1 out of 6 patients, 1 out of 6 toxicities, we find that there is a 50% chance that we are underdosing. Okay. So as a clinician, when you see this, what would be your recommendation? Your recommendation is, I think we need to escalate and move to the next dose level. Okay. Right. And that is what we do. Okay. So if you see now, okay, and I'm just going to skip a couple of slides. This is the sort of information that we supply to the clinicians, and the clinicians and statisticians sit together and say, now let's look at these at this particular dose level. Okay. So at dose level 1, okay, and the lowest part is your underdosing. The lowest graph is underdosing. The middle one is targeted toxicity. Okay. The top one is overdosing. Okay. So probability of underdosing at dose level 1, 2.5 is very, very high. Okay. So we want to go up. The probability of targeted toxicity, okay, in all of these lower doses is almost negligible. Okay. The probability of overdosing as we go to the higher doses is very, very high. Okay. So what we want to do is we want to look for something over here where we are not in red, we are still in green, okay, in overdosing. Okay. Our targeted toxicity is the highest and underdosing is as low as possible. And that happens at dose level 15. So based on the current information, I would say the recommendation would be that the next dose level we should go to is 15. Okay. Now, once we start to put a number of patients there, we can put 1 patient, 4 patients, 5 patients. Okay. Once we start, we can even start looking at 2 patients or 3 patients. You can look as often as you want. As you start to collect information on dose level 15, these plots will change. Okay. So these plots are done for every time you look at the data. And then every time you either take the dose level higher or the dose level lower based on what this tells you. Till you get to a point where you can't go any higher and you can't go any lower and you have achieved optimal. And that is when you stop and say that is your maximum tolerated dose. Okay, right? So this is the kind of adaptive phase 1 designs that we now use in some of these more critical settings where we have to find the right dose in a phase 1 setting itself. Okay. So what are the pros and cons? Very quickly, I have to move to the next part. But so between an algorithmic and model-based, the algorithmic, as you saw, is very, very easy. The model-based is a little bit more complex. But then the algorithm is not very flexible. Model-based allows you a lot of flexibility. Okay. It actually gives you a lot of information about the true DLT rates. You can optimize your model. You can add things to it. Right now this model was based only on safety. You can add efficacy to it if you like, and so on. Yeah. And so the recommendation these days in most of the companies is when you look at some of these rare mortality kind of indications like oncology and so on, you typically go for model-based design versus the algorithmic designs. Okay. Good. So we did the summary. And if there are no further questions, I'm going to go to phase 2 just to give a quick sense of what we do in phase 2 settings. You can take a short break to see if there are any questions that you want to talk about.
How many observations do we need to find out that this is the correct a prior information? There you said 1 out of 6, but isn't that number too small to use for a model?
No, actually, 1 out of 6 is, so 6 patients in a cohort is very, very good already, because, you only are looking for one parameter, no binary parameter, yes or no. Right? So it actually starts to give you a lot of information. But the best part about these Bayesian designs is it's not only the information from that cohort that you take information from all different cohorts as well, correct? So you basically say that, my model gets improved by saying I have no toxicity at 1 milligram, no toxicity at 2.5, 0.16 toxicity at 5, and so on. So you're taking all that information in your model, okay, so that over the course of time, your model keeps on getting better and better as you go to the higher doses. Yeah, so sometimes you don't even need 6, you do with 4, you do with 5. There is no fixed rule anymore.
Regarding this Bayesian design, is it frequently used only in phase 1 oncology trials, or is it frequently used only in oncology trials, or is it used in non-oncology trials?
You can use it in non-oncology as well in very, very rare, kinds of cases. Like, for example, if it's a rare indication with mortality, right? You can use, or not just mortality, but significant, debilitating disease, you can use it in any of those. So for example, we use designs like this even in snake bite, for example. Right? I mean, we're using a 10+10, but we can use a BLRM type of design. I know Dr. Jaiseelan was considering a BLRM type design as well for the snake bite study. So any of those studies where you can actually, where your primary, your dosing, your medicine is very, very toxic. Yeah. In any of those kinds of settings, and the outcome is death or something very, very close to death, advanced hospitalization, things like that. So you can use this in any of those where your primary method of fixing a dose is based on how much they can tolerate. Yeah, these designs work very well in those settings.
Regarding this 3+3, like how to decide whether it is 3+, because most of the literature says they use 3+3, but can we use 4+4, 5+5, 6+6? On what basis do you decide these numbers?
Yeah, so it depends. One good way to think about that is where your target toxicity you want it to be. Right, a 3+3 gives you a target toxicity of 0.33, okay, roughly about 0.3, okay. So if you want your target toxicity to be 0.2, then you might do a 5+5 or something, yeah. So it all depends on where you would like your target toxicity to be. Yeah, good. Oh, one more.
Sir, in theoretical part, based on logistic regression, is there any particular family of distribution?
I'm sorry, is there any?
Particular family of distribution, like a normal or beta in terms of alpha comma beta?
Right. So I think the question you were asking is there a particular family of models that you use? Most of them that I've seen, because it's yes or no, right? Your DLT is either a yes or a no. You tend to use a binomial, okay? And so your prior, you tend to use a beta because it's a conjugate prior, right? So that's what you end up using and a Bayesian logistic regression is exactly that, okay? But you could use other designs where, instead of a logistic model, you could put a different model. If you think that your dose toxicity curve is not logistic in nature, you could put a different, like an Emax design, yeah? So for example, your dose toxicity could be Emax and not logistic. And then you use an Emax design instead of a logistic design. Yeah, and it's not only phase 1, by the way. I mean, we'll see something similar now in a phase 2 setting, in a non-oncology setting. We use similar kinds of these kinds of designs, and I'll just speak about that shortly.
There are no more questions.
Okay, excellent. All right. So moving on to phase 2, the whole idea of phase 2, we try to think about it as a phase 2A and a phase 2B. Yeah. And, essentially what they are is in a phase 2A, it is to establish what we call proof of concept. Okay. And proof of concept is to say, does the drug work? Yeah. Does it work in the way that we expect it to work? Does it hit the right biomarkers, for example, or does it give the right efficacy parameters? Right? Does it work? Proof of concept is essentially that, right? And a phase 2B is to confirm that and provide the setting for a phase 3. Is how many patients do you need in a phase 3, right? What is your therapeutic window? What is a good, dose level? Are there multiple dose levels? What is the right kind of dose level? And what should you go with in a phase 3 setting to maximize your chances of showing that your drug is better than anything else that is on the market? Okay, so this is how you separate your phase 2A and your phase 2B. Okay, so phase 2A is primarily to establish proof of concept. Does the drug actually work? Phase 2B is to get all the other parameters. What's the right dose level? Is it hitting the right biomarker? Is there a subpopulation of biomarkers where it's more effective? What is do you have enough confidence that the drug is working to take it to a phase 3? Right? So that's typically how you think about phase 2A and phase 2B. Okay. So we'll rush through the first few slides because they're very, very simple. These are very common though, unfortunately. These are designs which are based on what we call the width of a confidence interval. Okay. So you typically, there is no sample size justification at all. You simply say, okay, if the true response rate is 0.3, then I put 50 patients and 50 patients. With 50 patients, the maximum width of the confidence interval will be no larger than something. Okay. It's just a statement. It's got no statistical justification to it. Yeah. Many, many years ago, me and a colleague tried to put some more stringent stuff to it, basically to say, if your true response rate is 0.3, and let's say the competitor is already at 0.2, you want to have a confidence interval that excludes 0.2. Yeah. So at least use that to provide some sort of a justification for the sample size, right? So we did some work in that space many years ago. Yeah. But essentially, these are still very common, mainly used in a phase 2A setting where you're trying to establish, proof of concept. You're estimating what your treatment effect is. Is your treatment even marginally okay, right? So that's the kind of designs. Okay. Then you have more popular designs called like the Gahan design. It's what we call a 2-stage design. Okay. In the first stage, you put exactly 14 subjects. Okay. It's a fixed design. It's not like 9 subjects, 10 subjects or anything. You put 14 subjects. If you see at least one response, then you add more subjects to stage 2. Okay. And how many more subjects on phase 2 depends on how large what is your expected treatment effect? Okay. So if your expected treatment effect is, let's say 0.2 or 0.3, okay, then you might add 9 or 10 more patients. Okay. If your expected treatment effect is 0.5, then you might want to add fewer numbers of patients and so on. Okay. So how many patients you add in a phase 2 depends on what is the size that you're looking for. Okay. The issue here is, so basically you use it just as an estimate to figure out what your treatment effect is and put a confidence interval, same as the first kind. Yeah, has a major drawback that even if your response rate is very, very small, is as low as 5%, okay, the probability that you get one response out of 14 is more than 50%. Okay, so it is very hard to stop at a phase at a first stage. Okay, you almost never stop at first stage. You almost always go to a stage 2, and then stage 2 is where you add enough patients to basically get a sense of what your confidence interval should be. Okay. So Gihan and the first kind of designs are designs which are in a phase 2A kind of a setting. They are still used sometimes, but people have moved on, and we have another really, really popular design called the Simon two-stage design, especially in the oncology area. This is a amazingly popular design still. Yeah. And what this design does is used to rule out an effect of lack of interest. Okay. So let us say that you're developing a new drug, okay, and you want the response rate to be at least 0.2. Okay, you expect that your drug has a response rate of 0.3. Okay, 30% people respond. But if it is less than 20%, you don't want to continue on with the drug at all. Okay, in that kind of a setting, you use this Simon two-stage. Okay, and the Simon two-stage, the way it works is, let's say you enter N1 subjects. Okay, N1 meaning some number. Okay, let's say 10, 12, 14, 18, whatever. N1 subjects in stage 1. Out of that, if at least R1 respond, then you proceed to stage 2. In stage 2, you enroll an additional N2, and then you do your hypothesis testing at the end of phase 2. Okay. If you don't get a total of R responses, you reject. Right. And what these numbers are, N1, R1, N2, R2, they're all based on the parameters that you provide the design. What do you want your type 1 error to be? What do you want your type 2 error to be? What is your response rate of interest? Okay, you give all this information, and what is your total sample size that you're willing to spend? Let's say 100 subjects. You don't want to have a study more than 100 or more than 60. If once you do that, what it does is it optimizes in that space. It's a numerical optimization problem. It optimizes in that space and gives you what should be the value of N1, R1, N2, and R2. Okay, so that's what the Simon two-stage design is. It's a two-stage design. It works reasonably well, yeah, and is very, very popular. Okay, becoming less popular now, primarily because many of the new drugs, even in the oncology space, are not cytotoxic. Okay, so you don't get a response rate immediately. What you get is that the drug cuts off, it works on the immune system, or it works on the pathway. It cuts off, funding, or, it cuts off whatever, thing that you need to give for the cancer to grow. Okay. So it cuts all of those things off. Yeah. And then, so the tumor just doesn't shrink. It just doesn't grow. Okay. And then over time, it starts shrinking because you're cutting off the supply route. Okay. So that's how many of these drugs these days work. And because of that, these responses are not seen very often unless you consider stable disease also as a response. Okay. So it's still used, but not as often or as frequently as it used to be used in 10, 20 years ago. Yeah. Okay. And then there are many extensions to this two-stage design. Okay. Some where you stop early, when you see a positive result in stage 1 itself. Some where instead of 2 stages, you have multiple stages, stage 1, 2, 3, 4. Yeah. Some where you have what we call randomized non-comparative design. Okay. Where in each treatment arm, you do Simon two-stage or multi-stage in each of the arms. And you take the arms forward, the ones that work. Okay. So these are all extensions to this two-stage design. Okay. But a lot of them are very, very useful as well. Right. We move to this design that we call a randomized discontinuation design. So it tries to use the benefits of randomization. Okay. But what it does is it's very interesting. In stage 1, you take all the subjects and everybody gets a treatment. Okay. And then you take only the responders, and in the responders, you then in stage 2, you randomize them to treatment and placebo or treatment and control. Okay. And then you try to estimate what is your actual treatment effect for people who are getting a treatment arm over the placebo arm or the control arm. Okay. It's a very interesting design because what it does is it gives you a very, very good, positive estimate of what your treatment effect is. Okay. The only problem is it's too good. Okay, because you have already started with only patients who are already responding to your treatment. All the people who are not responding to treatment, you've already removed them. Okay, so the estimate of the treatment effect you get is very, very biased, upwardly biased. But again, it's used as a phase 2A design, proof of concept. Does your drug actually work? Yeah. So to answer that question, this works really well because you need very few patients. The sample size typically for these studies are very small, right? The other place where it doesn't work is if the treatment is curative. Okay, if you have an antibiotic study, if you give stage 1, everybody is already cured of the infection, then in stage 2, what is the point of responding anybody to anything, right? So if your stage 1 treatment is itself curative, then you can't use this design, of course. Yeah. Okay, but otherwise, it's a nice phase 2A type of design to see if your drug is working. Okay. Good. And this is the part I was talking to you about, which is called, these are some of the more interesting designs in phase 2. So the first few ones, I just added them in there for completeness because you're going to read, when you read papers, you'll see them. So you need to know what they're about. But these are the new types of designs called MCP mod. This was developed at my previous company at Novartis, I think in 2004 or something. Okay. And it uses the same concept like we had the BLRM type designs, but it uses in a phase 2 setting. And it actually has 2 steps. Okay. The first step is called the MCP step, the multiple comparison step. Okay. And the second step is called the modeling step. Okay. That's why it's called MCP mod. Okay. So MCP step is to establish a dose response signal, to estimate what your dose response is. Okay. So you say that for now, let's say I have 6 possible types of dose response curves. Okay. So it says which among those 6 is the most likely dose response. Yeah. So the MCP step tells you that. Okay. And then the modeling step comes in the second thing and says, now that I know which kind of dose response, I'm going to model it and I'm going to start getting these targeted doses of interest. What is my ED50? What is my ED90? What is my MED, most effective dose, right? What is the best dose I should go with? Okay, so that's how these MCP models work. Okay, so here is an example of how this works. Okay, so you see that this is the trial design. Okay, during the time of trial design, you say, I have these 6 candidate models of how my dose response can work. Okay, this first one is called the Emax design. Very simple, right? There is absolutely no effect for very low doses. Very quickly, it hits an effect and then it remains flat. Okay. So my therapeutic window where the drug works is very small, only from here to here. Okay. It works here as well, but it's flat. Beyond that, there's no point in increasing my dose. Okay. This is a little bit more, this is still the Emax design, but this is a little bit more gradual. So I have a longer therapeutic window. Yeah. So my response gradually increases as you go along. Okay. This is a little bit more logistic. Okay. So nothing then increases rapidly and then plateaus out. Okay. This is linear, which means as I keep on increasing the dose, the response keeps on increasing. Yeah. This is what we call an umbrella shape, which is as I increase the dose, my response increases, but beyond a certain point, my response drops. Why? Because it's too toxic and patients are dropping off and they're not taking the dose anymore. So it's probably too toxic. So this is what we call an umbrella shape design. Then we have the exponential, which is it increases very gradually in the beginning and then suddenly shoots up when we get closer to the right dose. Okay. So these are, let us say, 6 candidate models. Okay. For me, in terms of, and this I am thinking at the trial design stage. So at the trial design stage, I already say that I'm going to evaluate these 6 models or 7 models or 8 models, how many ever. Okay. And then I start looking at the data. Okay. And then at the analysis stage, I do this 2-step one. One is that I say which of these designs is the best design. So that's a multiple comparison procedures. Okay. You will hear a little bit more about multiple comparison procedures later on and why they're important. Okay. But this is the MCP stage where you say that out of these 6 designs, this is the best design based on the data I've seen. Okay. And then once I get that this is the best design, Then I start my modeling stage where I take the selected model and then I fit the data and I try to see what are the different ways in which. So this is actually the shape of my dose response curve. What you see below and top are the confidence intervals. Okay. And I start modeling the data and I start estimating my parameters. What is the best dose? What is the minimal effective dose? What is the maximal effective dose? And so on. Okay. Make sense? Yeah. Good. So those were the MCP mod designs that are extremely useful now. And in fact, the MCP mod was one of those things which the European Health Authority has actually, what they call, I forget the exact term, but it's kind of validated the model or something. So in a way, they have given a positive opinion saying that they feel that this particular model in a phase 2 setting is probably the most, is the best current existing among all the existing models for you to get a good effect estimate of your dose effect and what is the right dose to go by. Okay, so that is a positive CPMP opinion that that model has achieved. Okay, so very popular design these days. Okay, and then we move to this last part where you have phase 2 designs which are based on your biomarker activity. Okay, so you have the traditional what we call enrichment designs where you enroll all the patients and you find out which patients are biomarker positive or negative, and only the ones which are positive you randomize them into new treatment or control, and the ones who are negative you get them off study. This works when you only have a sub-selection of biomarkers. I mean, only the biomarker positive patients, only the biomarker negative patients are responding to your drug. Okay, and we have a few examples of that. Herceptin, for example, right, is one example where only if you're HER2 new positive, the drug works. Okay, same thing with cetuximab and EGFR, right? Only if you're EGFR positive, cetuximab works. Yeah, so all of those kinds of drugs can work better with these enrichment designs. Okay. Then you have what we call these all-comer designs, okay, where you enroll everybody, you split them into biomarker positive and negative, and then you randomize everybody into treatment and control. Okay. And here you can have two kinds of analysis that you can do. You can look to see whether biomarker positive or negative has an effect. So you can actually test new RX from biomarker positive to new RX from biomarker negative to get an estimate of whether the biomarker actually is a good surrogate, a good way for you to separate the patients out. Yeah. Or you can also look at the difference between treatment and control in both biomarker positive and negative to see whether your drug works in the entire population. Okay. So that's one way of doing it. The other way of doing it is actually flipping that. Okay. When you don't know which biomarker to look at, then you flip it and you say, I'm going to enroll everybody. I'm going to randomize everybody to treatment and control. And then amongst this, I'm going to start seeing how many people are, biomarker positive and negative, and how many people are biomarker positive and negative. Okay. So then here, what you would do is you would actually do a comparison between the biomarker positive for RX versus the biomarker positive for control. So you would do that comparison to basically see whether there is a separate treatment effect among the biomarker positive patients and a separate treatment effect among the biomarker negative. So this design also gives you an opportunity to identify biomarkers. Yeah, and typically these are the sorts of designs that you use in a phase 2B setting. Okay, and then one of the most famous type of designs in this biomarker kind of space is what we call the adaptive signature designs. This is again the same Richard Simon who put the Simon 2-stage design. He and his group have worked on these adaptive signature designs. And basically here what you do is you have a 2-stage setting, okay, a 2-step procedure. In the first, you randomize everybody to treatment and control, and you develop a classifier. Okay, a classifier here is a threshold or a biomarker or something. Any kind of a classifier you develop, okay? So you say that I am going to take only patients for whom the protein expression is greater than 30. Okay, that's a classifier. Okay, so classifier is what you develop in your stage 2. Okay, and you split your alpha between these two. So you give almost out of, let's say you have 5% alpha, you give 4% alpha to developing this classifier. Okay, And then in your stage 2, you again enroll a different set of patients, again randomize them all to treatment and control, and then apply the classifier here to see which are classified positive and classified negative. And then the comparison that you do is between the classified positives for treatment and control. Okay. Now this though only has 1% alpha left because you've already spent 4% in developing the classifier. Okay. So this is very similar to the kind of designs that you see these days where you have testing and validation, right? In all of these new algorithms, you basically do, develop your algorithm in the testing phase, correct? And then you test that in the validation phase with a different set of patients, correct? So that's the sort of, thinking here in having this two-step procedure for biomarker activity. Yeah. Okay. Good. A quick talk about screening designs. The screening designs are basically very, very late stage phase 2 designs. Okay, they're almost like a phase 3, but except that what happens is you say that instead of setting up a big phase 3, I'm going to do a smaller phase 3 with a bigger alpha. Okay, so what you do is that you have a very large, for example, your alpha, you increase it to 25% instead of 5%. Then instead of 380 events, you need only 190 events. You need only half the number of events. Okay, so it's almost like doing a smaller phase 3 study, and they just call it a screening design. And most of the time, these screening designs are sort of like a adaptive design. So you do the first part as a screening design, and then you look at what you're getting, and then you just add on a phase 3 component to the top of these designs. Yeah. So those are the kinds of screening designs. Okay. Now the last part that I wanted to close with, I'll need another 5 more minutes if that's okay. 5 to 7 minutes. Yeah. Is what are some of the main challenges with some of these classic designs? Is that the standard classic design, right? They are suited for testing this sort of null hypothesis of no effect, right? So you're basically saying I have no effect, okay, and you're testing that. Okay, that's what the classical designs do. Okay, but that's not what you're trying to do in phase 2. In phase 2, what you're trying to show is that I want an effect beyond a certain point. Okay, the comparator already has a 40% effect. I want to do better than 40% effect. Okay, so that is what you want in a phase 2 study, not is there any effect at all for my drug? The drug may have a 30% effect and may be statistically significant, but what is the use because the comparator is already better than you? Yeah. So what you want is you want to have a design that helps you assess if there is evidence beyond a point of indifference. Okay. And let us say that we call this point of indifference as theta. Okay. This is not your treatment effect. Okay. Please remember your treatment effect is delta, which is it better than 0? Okay. All right. Or it could be delta could also be if it is better than the control. Okay. But theta is indifference. Okay, so for example, let us say that you have a study in which the comparator effect is 0.4, okay, 40%, and your effect is 45%. Okay, but you do this, you go out to the industry, and the industry says, nah, what does it matter? 40, 45%, almost same, I don't care. Yeah, so nobody wants to buy your drug. Okay, so now you're saying that your treatment effect delta is 5, 45 minus 40. But your indifference point is, let's say, 10, which means that people will be excited only if you're better than the existing by 10. Okay, all right. So theta is your indifference point. Okay, and what you want in your phase 2 setting is to check if there is enough evidence that your drug is better than that indifference point. Okay, and so that's what you try and do here. If you look at this graph, okay, there are 2 things going on here. Your alpha of 22.5%. Okay, this is your standard classical thing. Okay, is your drug effective? Okay, which means in this case, let's say hazard ratio, you have not, seen hazard ratio, that will come later. Okay, but let's say your, proportion. Okay, is your drug effective? Is your response rate greater than 0? Right, that is your alpha, classical approach. Okay, but beyond that, this blue part essentially says, my hazard ratio, my log of hazard ratio here, I want it to be, is there at least a 50% chance that my hazard ratio is less than 0.75? Okay. Now your hazard ratio in this case, just a quick note about what your hazard ratio is. Hazard ratio is your estimate of your effect from a survival standpoint. Okay. So lower your hazard ratio, better your drug is. Okay. Hazard ratio of 1 means you're the same as competitor. Okay. All right. So when you say log of hazard ratio greater than 0, log of 1 is 0, right? So is your hazard ratio less than 1? That is what this red part is testing. Okay. That is your classical phase 3 setting. Okay. But what you really want here is to say, but is my hazard ratio actually less than 0.75? That is what I want to know. Okay. And that part is done by this blue thing here. So what you have is 2 criteria. Criteria 1 is, do you have evidence beyond a point of indifference? Okay. That is at least a greater than 50% probability that your log of hazard ratio is less than 0.75. Okay. And 2, is there statistical confidence that this evidence is real, that truly a hazard ratio is less than 1. So you have 2 criteria. And so you actually set up your study to be able to take care of both of these criteria. So your sample size and everything is based on these 2 criteria. And those are what are called the double criterion designs. Yeah. And these are the sorts of designs that a lot of companies tend to use in a phase 2 setting because they want to answer 2 questions. 1, does the treatment have any effect at all? 1. And 2, does it have the effect that I'm interested in? Does it have effect beyond an indifference point? And to answer those 2 questions, you need what we call the double criteria designs. Yeah. So I think that was the last thing I wanted to do, and I want to stop with that. Okay. A quick read through phase 2s. Phase 2s typically are, those kinds of designs that you use to get more evidence about whether you should go into a phase 3 or not and get the right dose level. The right dose level, typically, a lot of people tend to use MCP mods for that. And to get evidence on whether you should go into a phase 3 setting, you start to use these double criteria designs. Yeah, good. So with that, I'm going to stop and open it up for any questions.